Swayam Prabha. Digital India. Educated India.
Then we will explain few more parameters. One is a flexibility parameter, B factor. What is B factor?
The thermal index.
Yeah, you can see thermal fluctuations, right? How far we can accommodate right within this electron density map. So, this is the values. So, you can see this is x, y, z coordinates, this is occupancy, and here this is the B factor. So, you have different numbers in a protein, and if you want to compare this B factor with the different proteins, because it changes from protein to protein. So, in order to normalize that, we can derive the normalized B factor using the formula B normalized equal to B minus B mean, right. What is B mean?
Average of.
This average of all this, okay, this is the numbers. So, you can get the average. So, this is equal to B mean. Then we get the B sigma. What is B sigma? This standard deviation, you can see the deviation from all these numbers. So, we get the B sigma. So, we get this formula B minus B minus B sigma, right. We get the normalized values. If it is more than 1, we treat this as a flexible, and if it is less than 1, we take this rigid, we get depending upon this value of this B. If it is more than the mean, right, or less than the mean. So, you can see that it is flexible or rigid. With more than the mean, right, it is more flexible, right. If less than the mean, then it is rigid. So, using this formula, you can see which residues are flexible and which residues are rigid. So, this is a physical quantity. There are also several physical quantities. For example, radius of gyration, center of mass, moment of inertia, all the information you can calculate from protein 3D structures, right. So, one of the center of mass, what is center of mass? Right. This is the center of a distribution of a mass in space. It has the property is here, right, that the weighted position vectors related to the point sum is equal to 0. For example, if you have 3 different objects here. So, you can see the center of mass, right, is located at position number C. So, how to calculate the center of mass? So, you can use Cm, here this is for the any coordinates, x coordinate or y coordinate, z coordinate you can calculate. So, mi into xi, where is mi is the mass and xi is the coordinate, normalized with the total mass, right. If you use this formula, you can calculate center of mass for the different coordinate system, or you can see where is the center of mass for the whole protein. Why how far this will be helpful? Because when you compare the different protein structures, so small proteins or big proteins and all, and the distribution of the residues, right, in a different shell. So, you can see in this case it is better to find the center of mass where it is located, and based on that you can see how far each residues are located with respect to the center of mass. Then we can also derive radius of gyration. What is radius of gyration? It is a distribution, right, of the components of an object around any axis. For example, if you take the moment of inertia, right, it is the perpendicular distance from the axis of rotation, right, to any point mass, for example, say mass m. So, you can calculate the radius of gyration using this equation. I, this is the moment of inertia, m is the mass. What is the equation for the moment of inertia? I equal to mr square, right. So, m is the mass and r square you can calculate from the center of mass, right. X is any coordinate from the center of mass. So, x minus em whole square. Now, substitute these values in this equation here, then you can get the Rg, then is sigma mi, right, multiplied by xi into cm the whole square, divided by this m, this sigma mi, then take the square root. You can calculate radius of gyration. This can also you can tell the information regarding the different positions, right, for the atoms in any particular protein. When you discuss about the molecular dynamic simulations, right, how the atom positions vary, right, that you can determine using this term about the radius of gyration. Now, we discussed various aspects, then we can also use the distance between the different residues or atoms to convert into the type of interactions. There are various interaction in protein structures. What are the various interactions in protein structures? hydrogen bonds, electrostatic interactions, right, hydrophobic interactions, van der Waals interactions, right. So, really you can calculate the values from the 3D structures, and also it is possible to see the contacts to identify what are the probable residues which can make this type of contacts. How many residues in a protein which have the potential to form any specific type of interactions. Instead of doing the complete calculations, you can identify the residues or residue pairs which are involved in any specific type of interactions. For example, in the case of disulfide, here you take the distance. How to calculate the distance? Square root of x2 minus x1 the whole square plus y2 minus y1 the whole square plus z2 minus x1 the whole square. So, you can calculate the distance. Now, for the case of disulfide bond interactions, you can see the pair of cysteines, because these cysteines form the disulfide bonds. Which atom in cysteine? Sulfur, right. So, you can see whether any sulfurs which are within the limit of 2.2 angstrom, they are considered as a disulfide bridges. They have high tendency to form disulfide bonds. In this case, for example, if you have protein structure, right, how to identify the disulfide bridges, whether any disulfide bridges in a particular protein? We have the structures. First you search for cysteine, and the cysteine we know the atom position for the sulfur atoms, and you get the distance. If the distance is less than 2.2 angstrom, then you see that these two cysteines, they have high potential to form the disulfide bonds, right. These disulfide interactions. Then you can see the ionic interactions. Essentially, what is ionic interaction? Coulomb's law. It is the interaction, attractive interaction between charge residues, one positive charge, one negative charge. What are the positive charge residues in a protein? Arginine, lysine and histidine. These are positive charge residues. What negative charge residues?
Aspartate glutamate.
So, this is negative charge residues, right. Then you can see any pairs which are within the limit of 6 angstrom. So, in literature different distances have been used. For example, sometimes they use 4 angstrom, but in this paper they try to use 6 angstrom because they do not want to lose any of these ion pairs. So, can we consider all atoms within the limit of 6 angstrom in the arginine and aspartic acid or glutamic acid?
Only charge
Right. So, if we take the arginine, there is several atoms. Also, if we take the aspartic acid, glutamic acid, we have several atoms, right, main chain atoms, NC alpha CO and several side chain atoms. So, it also if you take arginine or lysine, you can see a NH2 group. So, they have the positive charge. Likewise, aspartic acid or glutamic acid, right, you can see OE1 or OE2, right, they have a negative charge. You get the distance between these atoms and check whether the distance is less than 6 angstrom. Especially in the case of lysine and arginine, it has long side chain. C alpha will be somewhere, and if you see this NH2 group, this will be very far, right, because several CH2 groups, right. So, in this case you see the distance between the positive atom and the negative charge atom, and the distance is less than 6 angstrom, this can be treated as, right, high potential to form ion pairs. Likewise, you can see the aromatic interactions, right. What are the aromatic rings in the protein structures? Tyrosine, tryptophan and phenylalanine. So, you can see the centroid of the phenyl ring. How to get the centroid of phenyl ring?
Center of mass.
Right, you can see the coordinates for all the carbon atoms in the ring, right, and then see the centroid, that means you can get the average values like kind of center of mass, you can see the centers for the different rings. Then check the distance from different rings, and if the distance is 4 to 4.57 angstrom, then you can see that these two residues which are involved in aromatic interactions, right. In this case, if you take a protein, look at these rings and then get the centroid and get the and calculate the distance, and the distance is 4.527 angstrom, then you can say these residues are involved in aromatic interactions. Likewise, aromatic sulfur interactions, here sulfur atoms of cysteine and methionine, because two residues they have sulfur atoms, but when we consider disulfide bonds, we consider only cysteine. If in the aromatic sulfur interactions, we take both the residues cysteine and methionine and the aromatic rings. So, they take the distance of 5.3 angstrom. Likewise, in cation by interactions, what is cation by interactions?
Aromatic and
What is cation?
positive charge.
Positive charge, right? Positive charge. Mainly lysine and arginine, right? Then the pi, what is the pi system? Phenylalanine, tyrosine or tryptophan. So you can see the interaction between the charged residues, positive charge residues and this ring systems, right. So if there is the distance of 10 angstrom, then you can treat this as cation pi interactions. In fact, in the original paper when Gullivan Doughty they developed the concept of cation pi interactions, right, this 10 angstrom distance is so long. So they imposed constraints with the interactions energy. They computed the electrostatic energy and van der Waals energy. How to get the electrostatic energy? Q and Q2 by R. So you can see the electrostatic energy, right. We will discuss in one of the later classes about the details. And the van der Waals energy determined by the Lennard-Jones potential, 6L potential, and get the energy. And the energy is also there some cutoff. For example, this is less than minus 2 kilocalorie per mole. Right. In these things, we are confident that these two residues are there forming the cation pi interactions. If we take only the distance, right, maybe they may be very far. So the energetically may not be favorable, but they have high tendency to form the cation pi interactions. When you really see the cation pi interactions, we need to consider the energy along with these distance criteria. So you can get the hydrophobic interactions. Here you can see the C beta residues, right, because Cl pi is the main chain. So we go to the side chains, we need the C beta. So this way they consider C beta residues of this specific hydrophobic residues, and they consider cutoff of 5 angstrom, and then see if the Cl bar C beta residues which are within the limit of 5 angstrom, then you can see that these residues are involved in hydrophobic interactions. Then go with the hydrogen bond interactions, right. What are the requirement for a hydrogen bond?
2 electronegative.
Two electronegative atoms, one with the hydrogen with the attached the hydrogen and the distance, right, that is the about 2.5 to 3.2 angstrom. So you can get a hydrogen main chain interactions, main chain side chain interactions, side chain interactions. You can see all the hydrogen bond distance, right, if you have the calculate the distance, right, between these heavy atoms. So we discussed about various types of interactions and various parameters, properties which you can derive from protein 3D structures. Doing computationally all the parameters requires time, right, and again there is the program called PDB param. This is an online resource for the structural parameters of proteins, right. This is for they use the PDB structures, this way they put PDB, they derive various parameters, this is why they use the term param. So PDB param tells you and provides you the numerical values for several parameters which can be obtained from protein 3D structures. If you go to the PDB param, so there are 4 different types of values you can get, right, and 4 different aspects. You can get the binding sites that I will explain later when you discuss about the binding sites, and you can see interresidue interactions. What are interresidue interactions we discussed?
Aromatic interaction.
Right, you can see the short range interactions, medium range interactions, long range interactions, and you can get this contact order, you can get the long range order, all these things you can get. The secondary properties, you can see the tendency to form helix, strand and so on, and the physical chemical properties, if you see there are various types of interactions, radius of gyration, center of mass and the different types of interactions, right, electrostatic, cation pi and the hydrophobic interactions and various type of interactions. If you are interested in the interresidue interactions, for example contact order or long range order or short range contacts or long range contacts, you go with the interresidue interactions. If you click on interresidue interactions, right, then you will get several options which parameter you want. So to decide these parameters, right, we also require the PDB file. So in this case, in the previous slide you can have an option, so to enter the PDB ID, right, here I gave the 2 LZM, right, either you can give the chain name or without chain name also you can give the PDB ID. If you give the chain ID, it will calculate only for the particular chain. If the chain ID is not specified, then you will calculate for all the chains. Either you can use the PDB ID or also you can upload the file, right, in the PDB format. So if you have your own file, for example if you obtain any structures from MD simulations, right, the structures are not available in the protein data bank. So if you want to calculate the parameters, then you can upload the file So, then you can get the parameters. So, both options are available in PDB param. So, now we give the PDB ID and you want to get some parameters, right. So, for example, the interrestive interactions, right, it tell you okay, this is the ID, PDB ID, you can check, and there are various options available, there are various properties. What is short range interaction? 2 or 3 residues, right, the 1 and 2 residues, and the medium range interactions 3 or 4 residues, right, and long range interactions more than 4 residues, right. In these programs, so they have a little bit difference in this limit that I will show in the slides. Then you can calculate the contact order. How do you get the contact order?
The number of contacts,
Right, number of distance separation normalized with the number of residues and number of contacts. Long range order,
number of long range contacts.
right, you can see consider only if the 2 residues are separated by minimum of 12 residues, right. Total contact distance is another parameter which is the combination of contact order and long range order. They combine these two and then develop this parameter total contact distance. Then also you can see number of contacts within 8 angstrom either from C alpha atoms or C beta atoms, and 14 angstrom C alpha and C beta atoms. Why they give 8 angstrom and 14 angstrom? Because these distances are widely used in literature. 14 angstrom covers almost several regions in a protein, and 8 angstrom is well used in several aspects. Then you can get the multiple contact index, right, for different types of proteins. If you want to get all the data, just click on all. Otherwise, if you are specifically interested on the long range order or contact order, you can get the data. So, if you submit, you will get the output. So, this is the residue number 1 is contact with 2 and 3, 2 is contact with 3 and 4, right, 3 with 5, 4 and 5. So, here this is the residue number, this is the chain information, this is your central residue. So, these are the contacting residue. So, for example, this one is contacted with these 2 residues, they are located in positions 2 and 3, and this is the distance, right. So, you can get all the short range contacts. Let us go with the medium range contact, right. You can see the distance here, they use the distance of 5 and 6, right, for the case of 2. So, they use 5 and 6, right, they use the I plus 3 and I plus 4. So, then you get these are central residue, these are contact residues, and you have the distance. Distance we give the cutoff of 8 angstrom, right. So, we get the distance of 8 angstrom, we can get the numbers. Likewise, you can short long range contacts. For example, if you see these residues 6, right, with the tyrosine 161, and see the residue 7, how many long range contacts for residue 7?
3.
Right, 3, right, these 3. For example, if you want to get the long range order, right, what is Nij?
3.
3? No, because we need to have the cutoff of 12 residues. So, this is the I and here this J, totally 3 contacts. How many of them have the distance separation 12 residues? 2, right, because this is 5 and this, right, this case Nij equal to 2, right, you can get these values, right. If you see the long range order, you can see the numbers. So, now we go with the contact order. So, for this case we have the number 9.102. If you do the calculations for some all alpha proteins and some all beta proteins, you can see the difference, right, how far the contact order, right, varies based on different structure classes of proteins. Then long range order we can get for all the residues. So, here the total value is equal to 0.744, right, and for all each residues you can calculate. So, for example, take residue number 1, long range order equal to 0.006 because we have one contact. So, you can see is one contact, in this case 2 contacts, right. So, this 7, this will be 0.012, number of residues equal to 164. So, we divide by this 2 by 164, you will get this number, right, you can, then this is fine. The multiple contact index you also can calculate, here the value is 0.018. Compared with the contact order, long range order and multiple contact index, you can see the number is in which order? Decreasing order. Why is decreasing order? Because we impose more constraints. First one, no constraint, the contact order they take all contacts and they give weightage to the distance separation, and long range order what is the constraint we give?
Greater than 12.
Greater than 12. So, in this case we reduce several contacts and you go with the multiple contact index, we again reduce because we see the residues which have at least 4 contacts, this way number is less. But if you have a set of proteins, then you can compare. Okay, on the second one, right, if you want to get different types of interactions or the center of mass or radius of gyration, in this case you select the physical chemical properties and if you submit, right, it will show you all the properties. There is various properties you can calculate, center of mass, radius of gyration, all types of interactions, disulfates, ionic interactions and several interactions. But several case I did not explain the details, but if you go to the PDB param, there is a file, right, the tutorial file, as well as you can see the compute file. There if you click on these files, you can get all the details, what is the criteria used for different interactions, right, as well as the formula and all the details you can get. So, here now if you have several physical properties, if you get to know all these things, you have put all, and if you submit, I show you 1 or 2 examples, right. This center of mass, okay, this is a molecular weight is 21 kilodaltons and this is center of mass for all the 3 coordinates x, y, z. Radius of gyration, okay, this is radius of gyration on the 3 different axes, and surrounding hydrophobicity, right. What is surrounding hydrophobicity? It is the average the total value hydrophobic values of all the residues which are occurring within the limit of 8 angstrom. See, if you see some residues, you know the contacts, right. So, you can calculate the values. In this case, for all the residues you have the surrounding hydrophobicity. For example, if you take some of the residues which are high, say 15 or 17, and some residues which are very less, say 9 and 10 and so on. Even some polar residues which are highly hydrophobic, if they are surrounded with the hydrophobic environment, hydrophobic residues. Non-polar residues also, if you are in the midst of the polar residues, they have less, but you take the average and then you can see the tendency of these residues, right, specifically between the polar and non-polar residues. So, now if you get 2 proteins, so we discussed about the various parameters, then we can also see how far the 2 structures they can be superimposed and how far they are similar to each other. Here I show 2 proteins, one is the lactalbumin and lysozyme, they are from different families. So, 2 different proteins, these have 123 residues and 129 residues. Sequence identity is less, but if you compare these residues, look into these residues, you can see some sort of similarity in structures. See these helices here, we can see some similar type of helices and the strands here, similar type of strands here. So, how far they are different structures? In this case, put one structure and put the other on the top and measure the distance between the different locations, right. This will give you the root mean square deviation. This will tell you how far these 2 proteins can be aligned in the structures. There are various methods to align the structures, for example, distance based methods and vector based methods. I will explain briefly about one method called the DALI, right, distance matrix alignment developed by the Home and Sander, right. So, it is mainly based on the contact maps. So, we discussed about the contact maps, it is a representation of the 3D structure information in 2D form. So, we get these contact maps and we compare how far the contact maps are similar. How to do this? First is set the subset of the residues, they have the hexapeptides cut into different pieces and then see how these hexapeptides match. So, for example, there are some helices in the N terminal in the first protein and the C terminal second protein, if they have same contact map or similar contact map, then they can be aligned together in 3D structures. They use the search using Monte Carlo algorithm, right, using the greedy algorithm, it will find most probable matches and then combine together and put it together in the 3D structures. How they do? So, for example, this A and A dash, if one is in A and the second is A dash, the same place, directly you can align. Something is the second is B and the B dash is very far, then you see the contact map and this can be aligned together, put it together. A second option is an overlapping pair, A B with B dash and C with the C dash, and the third one you can combine these 2, right. A is directly mapping, exact match, and here B with the B dash and C with the C dash. So, when we collapse and then make similar to each other, B with the B dash and C with the C dash. So, now they calculate the score, they have a score just phi, this is the distance of protein A and protein B between any 2 residues. Using this formula, right, they can calculate the distance between I and J in the proteins A and B, and they get based on the score they can match the residues, they can map the residues. Once they map the residues, then they align together, right, and see the RMSD and see the best possible match, and finally they put together the two structures. So, this is the server for the DALI. So, it asks for the proteins, one is this first protein 1, protein 2. So, then take this protein and finally it asks for the superimposition, get the similar structures and do the structure based sequence alignment. In this case they do not align the sequence wise, they get the similar structures, they align together. So, based on the structures they align together, and using this information they will construct this superimposition. If you see the red, the magenta one and the green one, right, the red one is aligned very properly, right, the RMST is around 1.4 angstrom, right, between these two protein structures. So, recently they developed the server, you can see the new server. In this case they already calculated the similar structures for several cases, and they developed a database for the similar structures, right. In this case you do not have to calculate again. So, if you see these two proteins which are available in the database, then you can easily extract these two restive proteins which are close to each other and so on. So, overall if you see the structure based parameters, what are the various parameters we discussed? Starting from the beginning, we start from the contact maps, solvent accessibility, buriedness,
transfer free energy,
reduction in accessibility, contact order, long range order, multiple contact index, surrounding hydrophobicity, flexibility parameter, we discussed plenty of parameters, right. Then after that different types of interactions, what are the possibility of the two specific residues which can form electrostatic ion pairs or electrostatic interactions or disulfide bonds, cation interactions and so on. So, then discussed about the program called PDB param. It takes the PDB ID and calculates more than 50 parameters, right. This can be useful for any project, and you can take different types of proteins and calculate the parameters and see how the parameters vary in different classes with different functions, and see how why these parameters are attributed for different functions. Then we discussed about the alignment, how we can align these different structures. In the following classes we will discuss about the structure prediction, whether we are able to predict the 3D structures from the amino acid sequence, or the applications of these parameters what we derived in these 2-3 classes, right, to understand protein folding rates or protein stability or protein interactions, some sort of applications point of view we will discuss in the later classes. Then we will explain about the development of algorithms and what are the factors we need to consider when you develop your own algorithm, for example, for if you want to do any projects and so on. That will end up with that type of analysis. Thank you very much.